Good afternoon. I'm Don Giddens. I'm the Yellowstadt Chair Professor of Bioengineering in the Wallace H. Coulter Department of Biomedical Engineering at Georgia Tech at Emory. I'd like to tell you a little bit about some of the research going on in our laboratory today. We have the BioFluid Dynamics Lab located at Georgia Tech in collaboration with Emory. We focus on fluid mechanics, but our methodologies include biomechanics, imaging, vascular biology, and we're very interested in translation of our results to patients. Some of the current projects going on today are hemodynamics and atherosclerosis in mice. We're very interested in mouse models of atherosclerosis and in understanding the fluid mechanics and the interactions of fluid mechanics with vascular biology in mice. We have a second project on the relation of wall shear stress to plaque progression and to plaque rupture in human coronary arteries. We've just initiated a study of fluid dynamics of cerebral aneurysms and the relationship of fluid dynamics to complications of aneurysms and their potential rupture. We have a rather basic project going on with regard to flow field effects on the convection and attachment of nanoparticles in arteries. And we have a project going on in the liver, an organ a little bit different than our traditional organs of study in the heart and in vascular biology. So I'd like to talk a little bit about each of these, but first, some background with regard to atherosclerosis. There's a great deal of interest in the so-called vulnerable plaque. Virtually all of us, once we pass a certain age, have symptoms or experience atherosclerosis. The pathways of atherosclerosis can vary. The top pathway shows a very quiescent, relatively asymptomatic as one ages pathway of the disease process. On the other hand, often people will have advancing atherosclerotic plaques. The lumen available to blood flow in the artery becomes more and more constricted until blood flow is reduced and significant symptoms can occur that would require some form of treatment. But the most dangerous pathway is that prior to any symptoms, atherosclerosis is advanced and then suddenly a plaque ruptures, causing a catastrophic event such as a heart attack or even sudden death. Now, not only are we interested in the vulnerable plaque, but underlying all this are the risk factors associated with the vulnerable patient. So while we're trying to understand atherosclerosis from a mechanistic and biomechanics point of view, there's an awful lot to be gained from trying to avoid some of the things that make us vulnerable patients. We see the effect of fluid mechanics in many aspects of nature. Here is a river that shows a separation or circulation region where debris, if you like, can occur. On a larger scale, if you'll notice the winding path of this river, and the way the water flows, it's skewed to the outer banks, and you get a relatively low shearing area here on the inner banks, and notice the accumulation of sand and sandbars and so forth on the inner curvatures. While the arteries are at a very different Reynolds number than rivers, nonetheless, some of the same phenomena occur in our blood vessels. Our research generally follows the methodology of trying to examine individual subjects, whether it be individual humans or individual animals. We use imaging modalities in order to develop the anatomy of vessels so we can construct a geometry. Here, for example, is a human aorta that has been reconstructed from a series of images taken by magnetic resonance imaging. We can also determine velocity distributions on sections non-invasively in many cases, and also take a look at the compliance and motion information of vessels. Once we have a model constructed that is a computational model, we can apply principles of fluid mechanics in order to compute quite detailed flow structures in actual vessels. We can cross-check this with the experimental data, for example, by using phase contrast MRI on individual subjects We can compare measurements of velocity profiles by MRI with the computed values that we have from the computational modeling. So we're able to validate our computational models. Then this can be applied in a variety of areas, such as atherosclerosis in coronary arteries, studies of mice, and so forth. Now here is an example from the hemodynamics in atherosclerosis in mice study. This is an image taken from a mouse aorta. The diameter of the mouse aorta is only 1 millimeter, so this is quite small a vessel. We're able to strip away some of the superfluous vessels and then reconstruct this into a computational model. Here's an example of such a model that was reconstructed. We computed the flow field, and it's a pulsatile flow. Things vary with time and with space. This particular figure shows the time averaged wall shear stress in the aortic arch of this particular mouse. The hot spots indicating higher values of wall shear stress in dynes per square centimeter, and the cooler colors indicating areas of low wall shear stress. We tried to relate these biomechanics phenomena with measures of atherosclerosis. One of the biomarkers for atherosclerosis is, for example, VCAM expression. This is a vascular cell adhesion molecule. And these molecules are expressed in order to help capture monocytes in an inflammatory process. We can see from this study that there is a much higher expression of VCAM in areas where the wall shear stress is relatively low. And this undergirds the hypothesis that atherogenesis can be related to low areas of wall shear stress. We also look at human studies. This shows the methodology in a long-term study of plaque progression in human coronary arteries. Patients who have been referred to angiography because of cardiac symptoms go into the angiography suite, illustrated here. We have a calibration table so that we're able to locate the dimensions of the vessel in laboratory space and not just in the frames of the angiography. Here is an angiographic picture of the heart, blood vessels in the heart, that shows where blood is going. We are also, in this series of studies, able to go in with an intravascular catheter using ultrasound on the tip of the catheter. And this can be withdrawn slowly in the vessel to create a series of ultrasonic images with very good spatial resolution. From these images and from the reconstruction, we can develop models of coronary arteries in individual specific human subjects. We can then apply measurements of flow taken by the intravascular ultrasound system along with the geometry in order to compute flow fields in human vessels. This is an example taken from a human coronary artery. And in this particular case, you can see the geometric reconstruction of the artery wall. The color codes are where the flow is going in the blood and again represent wall shear stress, with the hotter colors being higher values of wall shear stress. And here you can see a cross-sectional image taken from the intravascular ultrasound of the disease that's occurring in this vessel. All of the green, white, and red here represent a rather large atherosclerotic plaque that has the potential to either obscure blood flow at one level or perhaps to rupture and totally occlude blood flow. We've recently initiated a study of fluid dynamics in cerebral aneurysm using similar methodology. We can use CT scan, for example, in order to reconstruct the anatomy and create a computational mesh. We can apply computational fluid mechanics to look at particle path lines. Here you see the aneurysm in this particular subject, and with the computations indicating where blood is going, where it's traversing, and also where it can linger for a long period of time. And here are computations of wall shear stress. The idea here is to try to understand what biomechanics forces contribute to both the initiation of aneurysms and also to their complication. The liver project is moving into a different organ, as I mentioned earlier, and there's been actually very little detailed hemodynamics work in the liver, although it's really full of blood, so many vessels going in and out of the liver, and with a great deal of variability depending upon the individual's anatomy. We're able, with contrast, MRI, PC-MRI contrast, magnetic resonance imaging, to get both images and blood flow conditions going into, this case, the portal vein. This is the superior mesenteric vein, the splenic vein feeding into the portal vein, and it bifurcates, in this case actually a trifurcation, into vessels feeding the right and the sides of the liver. There's been a good bit of interest, for example, in what happens to the mixing of blood as it comes from the superior mesenteric vein and through the splenic vein into the portal vein. This vessel, the SMV, tends to carry what radiologists call dirty blood from the gut into the liver, and it's thought that there may be some relationship between where this blood goes, it might contain toxins and so forth, to the development of liver disease in either the right or the left sides of the liver. Here we see where blood actually goes as it traverses from the SMV and the splenic vein into the portal vein. There's a great deal of swirling that occurs. This is all done by computational fluid mechanics, but we can trace then where the SMV blood goes, whether it's predominantly to the right side or to the left side, and try to relate this to the progression of liver disease. We have a number of collaborators in the projects. We work very closely with people both at Georgia Tech and at Emory. Just to list some of the major collaborators and a few of the students, Jen Su is in our laboratory in biomedical engineering. We have collaborators in DME and cardiology in several cases, as you can see here, with Dr. Joe, Dr. Taylor. Dr. Samity is an interventional cardiologist with whom we work very closely in the intravascular ultrasound project. We work with radiology very closely because of the MRI imaging that we use on individual patients. And of course, we have a number of students involved. Dr. Tong in radiology has just worked with us to initiate the cerebral aneurysm project. Stephanie George is a postdoctoral fellow who works on the liver project. Yi Gao is a PhD student who does image processing and image segmentation. Sung Ho Kim, a PhD student who is looking at computational fluid mechanics and solid mechanics. We have several funded projects. We're always interested in trying to add people to the group, particularly students and postdocs, but it's a very viable, active laboratory that tries to focus on both basic disease mechanisms and also translating both diagnostics and therapeutics into